As organisms, plants really are incredible. I've always found it odd how some people are just diametrically opposed to plants. Can't keep one alive, don't care about them, don't want to know how they work, don't really enjoy eating them that much either. They're amazing biochemical machines, and yet they're often overlooked because of how pervasive they are. Our whole lives are built around plants, even if we don't realize it. If you think about it, plants are essentially the ultimate way to turn air into food. I literally can't think of something more useful. It's often said that biology today is where computers were 30 years ago, and I think that's a very accurate description and provides a useful metaphor. There's a reason why many biologists stick to one type of organism. When you go from, say, animal biology to plant biology, it's like switching from coding in Python on Windows to coding in assembly on Linux on a Dvorak keyboard. Yes, you're still working on a biological organism, but very little carries over other than the extreme basics. You're still using DNA, cells still need to eat, breathe, release waste, and divide, and many core features are present, but even their execution can be very different. For example, the use of transposable elements, pieces of DNA that can cut themselves out of a sequence and jump to a new location, is way more common in plants, as is having weird numbers of chromosomes or polytene chromosomes like in the fruit flies we looked at in an earlier video. And unlike animals, most cells in plants are capable of generating an entire new plant from a small tissue sample without extensive biochemical help from humans. Just plate them on the right media and a small section of leaf grows into a brand new baby plant. There's lots of other examples, but I think you get the idea. As you can imagine, there's a lot to talk about when it comes to plants, from their biochemistry all the way up to their ecology. So today I want to start simple by teaching you a couple of techniques and then leaving you with questions that you'll need to answer yourself by exploring and doing the experiment. In an earlier video, we explored how a simple dye from a weird species of lichen paved the way for the discovery of the nucleus, chromosomes, DNA, and more. If you haven't seen it, I would highly recommend that one first, both because it's probably the prettiest video I've ever made, and also there are concepts we explored that might be helpful here. Link in the description. Today we'll be shifting our color palette a bit and using a different dye called Toluidine blue. It's an incredibly versatile dye that will stain all sorts of things based on the pH of the solution, and is used extensively in pathology and even has uses in surgery, though we won't be using it for those purposes today. This is honestly one of my favorite dyes, second only to Alcyon blue, which is used to make diphenized specimens and has a far more interesting history, so it's kind of hard to compete with that. Toluidine blue is a thiazine dye and is similar to methylene blue and several others. It's a much safer alternative to the Orcyon we used previously, but can stain chromosomes just as well. It's a very basic dye, which makes it stick to almost any acidic component of a cell. Another interesting property is that it stains different things different colors, which we'll explore in a moment. Today we'll be doing something called hand sectioning. Basically, this means using an incredibly sharp razor blade to cut little slices of plants that are hopefully only a single cell thick. Plants only have 3 organs, most of which is made of a network of tubes, so this will let us explore their internal plumbing and see a lot about how plants are organized and how they work. It's a tricky thing to do well and requires both having the correct type of blade and also a lot of practice. To be perfectly honest, the best images you'll see in this video were taken while I was in school doing this a couple of times a week in a plant lab. Right now, I am well out of practice and don't have the expensive microscopes that lab did, so the slides weren't quite as good. The setup for this is actually really easy. I'm working on a cutting board so I don't make a mess and have paper towels handy for any spills. I've got 3 little dishes here which I've filled with some water. Adding a few drops of 1% Toluidine blue to the middle dish and giving it a stir is all that's required to prepare our dye. You can adjust the pH to 4.4 with a bit of vinegar or hydrochloric acid, which will give better results and let you see the different colors, but isn't strictly necessary. The razors I'm using are safety razor blades. You want ones that are as thin as physically possible. Also, I would keep a few handy since we're going to be cutting through wood and they dull pretty quickly. We'll also need some little sticks to manipulate our plant slices through the washes. Snap the razor in half while it's still in the package and carefully remove one of the blades. Be extremely careful when you do this. I honestly can't explain how sharp these are sufficiently. If you get cut, the slice is so thin your body actually freaks out because it almost doesn't realize that you've been cut, so it takes forever to heal. Of course, we got the same warning when I started my plant class, and within 5 minutes someone had cut themselves, so be very careful. Now all we need is some plants to section. I wandered around my street to collect a few samples, and even took a few from my new planter boxes. Fair warning, the woodier a plant is, the more difficult this is to do, so start with something relatively soft like mint or basil, and work your way up to things like thin vines. Whatever you choose, it's best if it's fresh and used immediately after harvesting from the plant. But if you're going to need some time before starting, you can just let the plant sit in water for a while you set up. Honestly, letting a piece of stem sit in a dish of water for a few minutes will actually go a long way to make this easier. There are only really three types of sections that we can cut: cross-sections through the stem or root, longitudinal sections through the same, or cross-sections of a leaf. We'll be starting with stem cross-sections. Wet the razor and then grip it with your thumb and index finger, and then place your pointer finger on the top edge here to support it. Hold the blade perpendicular to the stem, and then in a continuous slicing motion, begin cutting little plant discs with the leading edge of the razor. The motion we want is a continuous slice. Don't saw at the tissue or you'll risk ruining it, and don't bend the razor or your slice won't be flat. Most of the time, one of two things will happen when you take a slice. Either it's too thick, or the angle is wrong, so you end up with a wedge rather than a slice. So keep making slices until you get a few that look so thin they're almost transparent and the slice is nice and flat. They'll stick to the razor thanks to us wetting it earlier, so just swirl the blade in our first dish of water to get them to release. For longitudinal sections, we'll have to use the cutting board. This time, cut thin slices lengthwise from the stem or root by dragging the blade through the section. It's much more difficult to do, as holding the thin stem and slicing it can be tricky. I much prefer cross-sections for this reason, but you see different detail. And finally, for leaf sections, how you do it depends on how firm the leaf is. If it's super floppy, like say basil, lay the leaf flat and cut straight down. Don't drag the knife like before, as you're more likely to tear the soft leaves. For really firm leaves, like pine needles or rosemary, you can actually cut cross-sections normally. Using one of our little sticks, swirl the water gently so that you can see all the slices, and then use the stick to pick up a good one. Transfer it to the Toluidine solution, but do so gently so it doesn't sink. If it does, you'll probably never find it again. Let it sit for anywhere from 30 seconds to 2 minutes depending on the solution concentration and pH you used. There's no exact way to know this in advance, so make a few of these at different staining times to see which look best, and then use that time for the rest of your samples. After it's done staining, use the stick to transfer it to the second water wash. Some people do a rinse in 15% alcohol first, which can help develop the colors better, but I won't bother for now and we'll go straight for the water wash. Let the sample sit in water and prepare several more. It's always good to make a few of these, as even two that look good at our scale may not look good under the microscope, and there's no way to know this in advance. But that's it. See, it's simple, right? Now let's see what all that effort shows us. Transfer your samples to a microscope slide with a drop of water, add a cover slip, and then have a look under the microscope. Already you should see a whole bunch of interesting detail. It always reminds me of a cross-section of a deep water internet cable. You probably noticed that the cells in the middle look a lot thinner walled and larger than the cells near the outside. There's also lots of color differences and maybe some structures hanging off the sides. And there seems to be distinct layers. It's really a whole world of detail, so I'm only going to cover the very basics, and won't get into what everything does, as that's a whole university course, or 3. The middle area with the large, thin cells is called the pith. It's very soft tissue, often used for storage by the plant, as it can't do much else since it's far away from the sunlit layers. Then you have the vascular bundles. There's two types of tissue here: phloem, which shows up as pink, and xylem, which stains vibrant blue. The xylem turns blue because its cell walls are highly lignified, which means they're essentially turned into wood, which toluidine reacts with strongly. Xylem transports water and minerals up from the roots, and phloem transports sugars and proteins from the leaves down and distributes it to the rest of the plant. Plant cell types are usually grouped by their cell wall composition. The pith cells and most of the non-vascular cells in the stem are parenchyma. These are multipurpose cells that do everything from storage of nutrients and starches to wound healing and repair. They usually don't become very stained as they lack lignin, or they stain pink. They also have very thin cell walls. Near the outside edge we have parenchyma, which have thickened cell walls and add a lot of support and rigidity to the plant, especially in young plants. Their cell walls are just cellulose though, so while thickened, they don't usually stain blue because they lack lignin. Finally, lignified tissues like xylem are known as sclerenchyma, and they're the easiest to find since they stain vibrant blue. There's a lot that can be said about these tissue types, but the thing I find the most interesting is that we can actually see how the lignin grows. If we look at a longitudinal section and look at the xylem from the side, we see this distinct spiral pattern. By sectioning the stem at different stages of woodiness, we can see this xylem development process, and these spirals form. The final layer is the epidermis, which is usually 1 to 3 cells thick, and these cells secrete all sorts of things to protect the plant, though primarily waxes. These cells will often have chloroplasts, and if not, there'll be a layer of cells below the epidermis that are full of chloroplasts, especially in non-woody stems. Sometimes you find little hairs hanging off the plant, and these are called trichomes. Trichomes honestly are a video in and of themselves because of their crazy diversity. Unlike in mammals, these aren't just hairs. They're often oil-secreting organs that produce a wide range of substances that they either secrete all the time or store and release when broken as a defense mechanism. Or sometimes they're sillified and become tiny razor blades or needles like in stinging nettles. It's really a mixed bag. So now that you know this technique, what do you do with it? Well, for one, I left out about a textbook's worth of detail. The more you look around, the more weird stuff you find. Like, what are these little granules here? Why is this cell full of spikes? What's this tube if it's not xylem or phloem? And so on and so forth. Every time you section a plant, you're opening essentially a whole new world to explore, and I think the best way to really understand it is to use this technique, find some weird things for yourself, and try and figure out what they are and how they work. To get you started, here's one example of a question that can be answered by this sort of experiment that I posed while I was taking my plant class. I love carnivorous plants, but was curious where the digestive enzymes came from, as there's no obvious pores in a Venus flytrap leaf, and there's no gallbladder to produce them like in animals. So I brought a Venus flytrap into class and spent half an hour carefully sectioning the leaves. Sure enough, what I found was fascinating. Up close, it was impossible to miss the enzyme secreting organs. They grow as little cauliflower-shaped buds all over the inner surface of the leaf. On the back surface, you can actually see them, but they're completely withered away, so the plant must grow them on both sides, but kills the ones on the back somehow. While I was looking around, I noticed some other interesting details. The trigger hair had a definite separation in it, which makes me think that the lower cells are the ones that get compressed, so I wouldn't be surprised if they're full of calcium ready to be released. And you may notice that unlike the tissue we looked at before, there's those little granules practically everywhere. These are starch granules, which the plant uses to store energy. I wonder if the density of them is so high just because this is a leaf, or if it's because of how energy intensive it is to suddenly close the trap of the plant. But this is what I mean. I got my answer, but now there's 1,000 new questions to be asked. So find a plant you think is weird and get to sectioning. Ask questions and poke around for the answer. Maybe somebody already knows it and you can look things up in the literature, but for all you know, you might stumble onto something new and weird. So that's plant sectioning, one of my absolute favorite microscopy techniques that requires almost nothing to try yourself. We'll revisit all sorts of plant biology and this technique in the future, where we'll be doing everything from growing plants in aquaponic setups to engineering their genetics to produce new compounds and grow faster. So be sure to check back every Monday for all of that. If you enjoyed this video and want more microscopy, I've already put out several other videos on the topic, including how to convert a cheap microscope to see polarized light, and of course the video about Orsian I mentioned earlier. If you're just interested in all the pretty pictures, then you may also be interested in my video on focus stacking, where I show how to create these amazingly detailed photomicrographs. And with that, I'll wrap up this video. If you enjoyed and want to see more, be sure to subscribe, leave a rating, and most importantly, ring that bell. This channel was made possible thanks to the donations of my wonderful patrons, and as always, I want to give a special thanks to all of you. As the channel has grown, your support has been the main thing that lets me keep the channel going and making these videos for you. So thanks again, and I'll see you next week.